Hello guys, it's Jacob Edwards here. I'm just going to be bringing you a quick video about the structure of DNA. Now the purpose of this video is of course to help you learn about the structure of DNA, because I'm going to be doing a lot of educational videos, and the first few videos I'm going to be doing is about DNA. So hopefully if you don't know anything about DNA, this video will help give you a little bit of hindsight going into the future videos. Now, first of all, we're just going to start off with what DNA stands for, which is deoxyribonucleic acid, which you can see right there. And when you think of DNA, you think of that twisted figure 8 type shape, right? That little wind. Well, that wind is called a double helix, which we can clearly see right here. And this double helix, there's a purpose. There's always a purpose for things when you're talking about science and biology. And a big thing here is structure equals function. That is a huge thing in biology. The way things are built, they're built for a purpose. Like the structure of DNA, it's twisted like this because DNA is, one strand of DNA is 6 feet long, okay? That's pretty long. And unwound, that's, pretty long. So it's twisted up like that and tied into actual chromosomes. And the reason for this is to make it smaller and more compact. That way it can fit through the tight places wherever it needs to travel. And DNA is made out of things called nucleotides. And nucleotides consist of a sugar, a phosphate, and a base. And this is very, very important because this is the whole structure. This is everything that DNA is made out of. So if we look here, we can see a better look of a nucleotide, and we can see that there is a sugar, there is a phosphate, and there is a base. Now, with bases, there's a rule, and this rule is that adenine has to pair with thymine, and guanine has to pair with cytosine. You will never see adenine with cytosine or thymine with guanine or vice versa. It is always adenine with thymine and guanine with cytosine. It's always going to be like that. I don't even think it's possible for it to be in a different way. Now, the things holding these bases together, what is holding them together? It's called a hydrogen bond, and this is very important. In the next video, I'm going to talk about DNA replication. And in that process, the strand of DNA will unwind and split. And the hydrogen bond is very important for the splitting because there are three main types of bonds. There's a hydrogen bond, an ionic bond, and a covalent bond. A hydrogen bond being the weakest out of the three, which means it's very flimsy, fragile, very easy to break, which is very good in this case. That way the DNA is easily separated in this process of DNA replication. Now, when we talk about the structure of DNA, something that points out is that it's anti-parallel. And what I mean by that is this side goes this way and this side goes this way. There is a 5' end and a 3' end on each strand. And if you look, you'll see why it's called anti-parallel, because when you think of something being parallel, you think of something being side by side to each other. Okay, well, this is actually crisscross from its matching pair. 3' is crisscross from 3', and 5' crisscross from 5'. That's why it's anti-parallel. They're running in opposite directions. Two lines going in the same direction, that's parallel. These are two lines side by side, yes, but they are traveling in different directions. That's why it's called anti-parallel. And that's pretty much it for the structure of DNA. Thanks for watching. I hope you learned something new from this video. If you have any questions, don't forget to leave them down in the comments below. I'll try to answer them. Don't forget to like and subscribe for future videos. The next video, like I said before, will be about DNA replication. So I hope you enjoyed, and I hope to see you all in the next video.